Hello, today we are discussing about the hydatid cyst. We received the specimen of the splenic hydatid cyst. So this black part is the spleen and this white part is the cystic structure. So the cystic structure covering almost all the part of the spleen. After the cut section, this is the cut surface, you can see this cystic structure enclosing almost all the part of the spleen, and inside the cyst you can see the granular material, membranous whitish part. These are the membrane of the cyst and these are the daughter cyst. These are the intact daughter cyst. They are very infectious also, and inside this granular material we can find this colles under the microscope. The cyst wall thickness is 3 mm. So this is all about the gross part of the splenic hydatid cyst. This is schematic diagram of the hydatid cyst. There are 3 layers. The outermost is pericyst. This green part is pericyst. This is formed by the compressed host tissue, and in this we see the fibrosis, and this is pseudocyst. Inside this there is ectocyst by the red color, and they show the laminated eosinophilic material under the microscope. The innermost layer is endocyst, means germinal layer, and here we can see the daughter cyst and scoliosis also, and inside whatever the material or fluid like is called hydatid fluid, and inside that we see the daughter cyst and the scoliosis. In short, about the life cycle of the hydatid cyst, it is a zoonotic or the parasitic disease caused by the Echinococcus granulosus or the multiloculus. The definitive or primary host are the dog and the fox. Definitive host means in which they gain their maturity or sexual stage is completed. They release ova or egg. The intermediate and secondary host are the human and the sheep. They ingest ova released by the primary host. So this life cycle occurs. The primary host ingests cyst and the secondary host ingests the egg. Now in human, through mouth they come into the intestine. This hydatid cyst come into the intestine, and from intestine they crosses the intestinal wall and reaches to the vessel, and finally through the mesenteric vessels reaches to the portal circulation, and we know via the portal circulation they reaches into the liver, and the liver is the most common site of the hydatid cyst, near about 50 to 70% of the cases, and they are the least common site. They are the lung, kidney and the spleen. Here we know today we received the splenic hydatid cyst. So primary splenic infestation can occur either via the escape of the liver lung barrier via the arterial route or through the retrograde portal circulation, because this is the portal circulation. This is portal vein. This is made by the spleen and the mesenteric vessel. If it has to reach to the spleen, there should be retrograde circulation. So this is splenic site is the least common. The other source of the splenic infestation is disseminated intraperitoneal spread. This I made the imprint smear of this hydatid cyst sand, and I found the hooklet. You can see very well the hooklet. This is refractile. Again one more, that is here, over here, refractile hooklet. These are the characteristic of the hydatid cyst. This section I have taken from the cyst wall. Here the arrow, you can see very well that this is pericyst. This is pericyst, and this is formed with the help of the splenic tissue. And here the blackish part, you can see the calcification also, and these are the calcification. And then this is eosinophilic part is ectocyst. So this is pericyst and this is ectocyst, and inside it we will find the endocyst. The arrow part, this is again ectocyst, laminated eosinophilic material, made up of laminated eosinophilic material. So this is ectocyst, and this is germinating layer, means endocyst. And this section, because this was very fragile in the nature, so inside the cyst cavity we have to find the squalecysts.